Bioinfocongress III Doktor Sinem Saka, moleküllerden dokuya, uzaysal biyoloji için DNA aracı. Doktor Sinem Saka, lisans eğitimini 2008 yılında Orta Doğu Teknik Üniversitesi Moleküler Biyoloji ve Genetik bölümünde tamamlamıştır. Doktora eğitimini 2013 yılında Almanya'nın Göttingen Üniversitesi'nde moleküler biyoloji alanında tamamlamış ve üniversite bünyesinde yer alan Uluslararası Max Planck Araştırma Okulu'nda Boehringer Ingelheim fonuyla doktora üyesi olarak görev yapmıştır. Doktora çalışmalarını yeni süper çözünürlüklü ve çok modlu görüntüleme yaklaşımlarıyla hücresel zarlardaki nano ölçekli protein organizasyonunun araştırılması üzerine gerçekleştirmiştir. Süper çözünürlüklü mikroskopu için yeni afinite problarının üretilmesi üzerine yapmış olduğu doktora sonrası çalışmasının ardından Amerika Birleşik Devletleri'ne taşınmıştır. Boston'daki Harvard Üniversitesi Weiss Enstitüsü ve Harvard Tıp Okulu'nda İnsan Sınırı Bilim Programı üyesi ve EMBO doktora sonrası araştırmacısı olarak yüksek düzeyde çoğullanmış görüntüleme yöntemleri geliştirmek için çalışmalarına devam etmiştir. Aralık 2020'de Almanya Heidelberg'deki Avrupa Moleküler Biyoloji Laboratuvarı'nda bağımsız araştırma grubunu kurmuştur. Burada hücre biyolojisi ve biyofizik biriminin ikincil bir üyesi olan genom biyoloji biriminde grup lideri olarak görev yapmıştır. Doktor Saka'nın laboratuvarı, uzamsal çoklu omikler için yeni etiketleme araçları geliştirilmesine ve uygulanmasına odaklanmaktadır. Aynı zamanda ölçekler arasında hücrelerin işlevsel durumlarıyla uzamsal ve moleküler organizasyonunun araştırılması için yöntemlerin geliştirilmesini hedeflemektedir.
Bioinfocongress III Dr. Sinem Saka, a DNA toolset for spatial biology from molecules to tissue. Dr. Sinem Saka received her bachelor's degree in molecular biology and genetics at Middle East Technical University, Ankara, Turkey in 2008. She completed her doctoral studies in molecular biology in 2013 at University of Göttingen, Germany, where she was a Boehringer Ingelheim Fund PhD Fellow in the International Max Planck Research School. Her PhD focused on investigating nanoscale protein organization in cellular membranes with novel super-resolution and multimodal imaging approach. After a bridging postdoctoral study on generation of new affinity probes for super-resolution microscopy, she moved to the USA and worked on developing highly multiplexed imaging methods as a Human Frontier Science Program, HFSP Fellow, and EMBO Postdoctoral Fellow at Harvard Medical School and the West Institute for Bioinspired Engineering at Harvard University, Boston. In December 2020, she established her independent research group at European Molecular Biology Laboratory, EMBL, Heidelberg, Germany, where she is a group leader at Genome Biology Unit with a secondary affiliation to Cell Biology and Biophysics Unit. Her lab focuses on developing and applying new labeling tools for spatial multiomics and methods to investigate the functional state and spatial and molecular organization of cells across scales.
Sayın Doktor Sinem Saka ve değerli katılımcılarımız, Bioinfocongress III etkinliğimize hepiniz hoş geldiniz. Değerli hocamız bizlere bugün A DNA Toolset for Spatial Biology from Molecules to Tissue konulu bir konuşma gerçekleştirecektir. Konuşma bitiminde ekibimizin hazırladığı ve katılımcılarımızdan gelen sorulardan oluşan bir soru-cevap bölümümüz olacaktır. Merhaba Sinem Hocam, tekrardan hoş geldiniz. Sizi görmek çok...
Merhaba Dilay, hoş bulduk.
Dilerseniz sunumunuza başlayabilirsiniz hocam.
Tamam, teşekkür ederim. O zaman ekranı paylaşıyorum. Her şey yolunda mı?
Evet, görebiliyoruz.
Tamam.
Teşekkürler.
Tamam. Sunumu İngilizce yapacağım ama sonrasında soru cevap kısmını tabii Türkçe yapabiliriz. Öncelikle burada olduğum için çok mutluyum. Umarım faydalı olur bu ufak sunum. Ben şu anda Heidelberg'de EMBL European Molecular Biology Laboratuvarı'nda grup lideri olarak çalışıyorum 2020'nin sonundan beri. Birazcık daha önceki çalışmalarımdan bahsedeceğim postdoc'tan ve biraz sonraki en son çalışmalarımıza bağlamak istiyorum. Şimdi İngilizceye döneceğim. Genel olarak, so our group is mostly interested in investigating molecular organization in context. And when we think of investigating in context, we can imagine, for example, this is our institute's logo from EMBL, as a biological sample, like a complex biological sample. And in red, we have, among all the other things, our favorite cell or molecule. The conventional approach would be to first label it in a targeted manner or observe or analyze it in physical or contextual isolation. But we really want to actually change this paradigm where we want to investigate it with context and learn details like who else it hangs out with, what's happening around it, how its state interplay with the surroundings. And of course, to do this, our favorite approach has been imaging. But going forward, we want to actually integrate this type of imaging-based methods with deeper molecular profiling methods to really get to the bottom of cellular states. This is primarily done currently at the omics-based levels, so using genomics, epigenomics, transcriptomic, proteomic, or metabolomic technologies. But there is a gap when we want to connect these to things like local and subcellular organizational features and their heterogeneity, which gives us information about spatial organization, cell-cell interactions, and similarly, things like protein translocations, moonlighting activities, and this new emerging concepts like cellular condensates and aggregates. This domain is typically addressed by more imaging-based methods, and we want to, going forward, integrate these two domains together much better so we can actually link those studies to each other. So realizing one of the biggest limitations is our tool sets and methods. I've been working on developing and applying new methods for labeling and detection of biological targets to be able to address some of the limitations and to also go all the way down to nanoscale organization, for example, including super resolution microscopy methods or correlative mass and imaging, fluorescent imaging approaches and probe development, also from my PhD onwards. And in my postdoc, I have been more focusing on repurposing DNA as an engineering tool to improve the resolution, multiplexing, and sensitivity of microscopy for spatial biology, using DNA-based imaging tools for example, single molecule localization microscopy like DNA paint or DNA exchange imaging. And we try to bring it all the way to tissue-level imaging as well. So, well, of course, when we talk about these method development, I want to highlight a few issues that imaging conventionally has, which affects our technical capabilities. And these were the problems that we wanted to solve with new approaches as well. So one of the important problems have been the diffraction limit, which actually limits the resolution of conventional imaging. And we can think of this as having, when the two objects we are looking at are closer to each other than the limit of diffraction, then we actually see them as one, as you see this in this example. For typical fluorescence imaging, this limits us to 250 nanometers or so in the XY dimension, and beyond that, we cannot really resolve things so well. But there are new approaches, and I'll talk a little bit about them. The other problem is the spectral overlap, which actually limits the multiplexing. So this is how many dyes you can put on fluorescently on molecules and actually still separate them well. And normally this is not too many. This limits you to 3-4 fluorophores at a time, so 3-4 different molecular targets. And we have a boundary to go beyond that if we don't use complex, let's say, spectral unmixing type of approaches, which have their own problems. So this is another issue that we wanted to tackle. Problem 3 would be autofluorescence, scattering, and dynamic range issues. So in this case, our sensitivity is actually low, and we also cannot see things as well when we have big ranges on the dynamic range. So let's say you have a few copies of a protein versus many, many copies, millions of copies of another protein. So it gets hard to use the same system to see them both. And in tissues, we have a lot of autofluorescence and scattering issues as well. And the last one is that imaging is usually, working at one field of time, and you only see what you mark. So for some discovery-oriented approaches, it is not the most suitable. And this also limits the throughput and content we can get from an experiment. So this actually creates major needs for new tools and techniques. And some of these methods, some of these areas have been really improved in the last decade. For example, the resolution problem, there was a Nobel Prize a few years ago for super resolution microscopy that actually several different methods tried to address this. And for the spectral overlap, recently there has been a bunch of multiplexing techniques that are addressing this issue. So I'll talk a bit about our solutions where we use DNA and repurpose it for these type of new methodologies, but there are also a wide range of other approaches that have been developed in this context in the last decade. So why do we use DNA? Because it is one of the simplest macromolecules out there. You know, it's only made out of four different components, and we understand it quite well. There is one key reaction where two complementary or partially complementary strands come together to make a double-stranded DNA duplex. And the parameters of this reaction is really have been understood quite well with decades of research. So we know that it's influenced by temperature, concentration of these strands, and other molecules or components like sodium, magnesium, divalent ions, formamide, additives, etc. So there are many computational tools that use this information to design DNA nanostructures or DNA reactions and extract the kinetic parameters. And these have been very useful for us to actually repurpose or engineer DNA. I mean, at the moment, using these tools, we can even create very complex, let's say, DNA origami or DNA nanostructures that can even be dynamic in certain aspects. But one of the features that we use the most is the orthogonal barcoding capacity of DNA, thanks to that unique combination of 4 bases, making it quite feasible to actually encode a wide variety of barcodes. So we have been essentially using this type of DNA toolbox or developing new approaches to address these limitations. I'll have the name of some of the methods here, and we'll briefly talk about them. So, for example, for circumventing the diffraction limit, we have been using an approach called DNA paint or expansion microscopy combined with our technique called SABRE. For multiplexing, we actually use a DNA exchange imaging principle. For sensitivity, we use a signal amplification approach we call SABRE for immunofluorescence and also for RNA DNA detection. And for getting higher throughput and information and actually bringing all the way to next generation sequencing, we have a new method called LightSeq where we integrate imaging and sequencing. So I will highlight a few of these going forward and also explain how we leverage them for spatial omics applications. So first, let's start with multiplexing and super resolution imaging. So basically, the way we typically use DNA probes is they become orthogonal labels. So if you were to normally label your target molecule with a fluorophore, you would put, using either directly or indirectly using like tags or antibodies, a fluorophore on it, and then use the imaging. But that would be resulting in the spectral overlap problem. So we instead uncouple the labeling from detection by using DNA-based reversible probes. So you have, again, your target molecule, but we kind of create a barcode on it by bringing a short DNA sequence that will be used as docking sites for the various applications. So in this case, for imaging, we can actually use a fluorescent oligo that will bind to this when we want. So it's an on-demand reaction and make it bright for visualization by microscopy. And when we are done imaging, we can also get rid of it because this binding is really DNA hybridization, and we understand the kinetics, so we can change the buffer concentration of, for example, salts or temperature or add formamide type of reagents to remove or to control the dehybridization in a programmable manner. And then we could also have orthogonal barcodes on multiple different targets. So we can add then orthogonal imagers, so these fluorescent short oligos, to detect them and visualize many different molecules like this. So this gives us a way to cycle between different targets and visualize many of these together without being limited by spectral separation of the fluorescent molecules. So this type of approach has been also used for super resolution microscopy. In this case, you can imagine we have, for example, like an RNA molecule that is detected by fluorescence in situ hybridization probes. And then we have these docking strands on the probes. and that these imager molecules can bind to those sites. So if we engineer this binding to be very transient, so it's very short-lived because the strands are very short, and they will bind and rebind, and we would see something like this blinking behavior. And one could actually look at the time course of these spots, so you can see binding dissociation, because there's spontaneous dissociation. And if you have the right concentration of imagers here, you would only have, in a diffraction-limited volume, one binding event. So you can say that, okay, I know this is coming from a single molecule, and I can actually fit the position of each spot because I know that will be a Gaussian curve. So I know with high accuracy that the center of this spot will be here. And even though the spot will be 250 nanometers, I can actually localize it with, 20 nanometer accuracy. And by doing then long-term imaging, let's say in fixed samples, you can generate tens of thousands of frames and then accumulate all of these spots so each molecule is detected and reconstruct the image from them. So this is a DNA origami nanostructure where we had 4x3 dots that are 20 nanometer apart. And you can see that we can reconstruct this image after imaging many thousands of frames based on this single molecule imaging. So this is the principle of DNA paint, but other types of super resolution imaging methods also utilize this position fitting for single molecules to generate similar high resolution data. Now, when we use orthogonal labels or DNA sequences on different molecules, in this case, we actually, in this method paper that was published by Ralph Filman a few years earlier, they have designed 10 different DNA origami nanostructures where the different numbers were kind of put together. So these are all the docking sites, and when you bind images, you would see different numbers appearing, all mixed together with orthogonal strands here. So you can actually switch between them and encode all of the 9 channels or 10 channels here by utilizing this type of approach. So this is called exchange paint, when we actually replace one imager with another and then re-image the same sample in cyclic ways to reconstruct the higher multiplexed image. We've also been using this type of approaches to get really high resolution images of, let's say, different molecules, proteins, RNA, DNA. In this case, we are looking at total genome, and we can see the areas of high density heterochromatin or the low density nucleolar areas or the ribosomal DNA strands, for example, with sub 20 nanometer resolution, both in X, Y, Z by using turf microscopy. I'll not go to too much detail here. This is more to also exemplify what type of applications are possible. So coming back to the exchange imaging, we can actually combine it with things like immunofluorescence to label a range of proteins. In this case, we would actually have the DNA barcodes and antibodies and apply them onto the sample at the same time, and then detect with these fluorescent oligos, one at a time or multiple at a time, depending on how many fluorophores we can use. So typically we could do 3-4 per round and do this sequential imaging for all of the fluorophores. But and some of the early applications, this was applied in things like mouse hippocampal neurons, where several different targets were detected. But one of the problems we have realized is that, of course, if you use secondary antibodies, you are quite limited by using mouse or rabbit antibodies because all the other species are not so available. So at some point, you need to label the primary antibodies, and that costs a signal loss. And especially for tissues, we realized that this loss of signal, which now you cannot really amplify by secondary antibodies, plus all the many fluorophores you put on it, is creating a problem. So especially due to autofluorescence and scattering in the tissues. This was a low detection sensitivity issue we wanted to solve again by using DNA. And people have been using really strong amplification methods typically. So immunohistochemistry, where primary antibody, secondary antibody complexes are used. Then the secondary antibody carries an enzyme like peroxidase or alkaline phosphatase, which converts a substrate to a color product. These typical images you might have seen in pathology slides are produced this way. These chemistries are typically single-plex, so you cannot look at many targets, but they provide hundreds of folds of amplification for robust detection in pathology samples. So we wanted to develop something that is a bit more, let's say, elegant, but would also allow us to do multiplexing and apply our methods in this context with high signal and detection sensitivity. So in this case, we thought, okay, we can go with the target molecule and now have a concatemer, so repeated units of the same DNA oligo strand, which would help us to bring many imagers onto one strand, so we can actually create an in situ signal amplification. So this is the principle behind the method I will talk about next. Of course, one of the biggest problems here is that how do we create those concatemers? It's hard to get them synthesized when you have a repeated sequence, especially when you have a reaction. So I'll try to explain this with an animation, but overall we have a short primer. This is a 9-10 nucleotide oligo, single-stranded, and then we have a hairpin molecule that has this overhang, as you can see, which is also single-stranded and complementary to the primer. There is also another feature in this hairpin molecule where we have this molecule called like a stopper site. And here the stopper is, we can think of it something that doesn't let the DNA polymerase to go through. So for example, it could be a C nucleotide where if you don't have any G's in the reaction mixture. So in this case, you would only be using 3 out of 4-letter alphabet in the DNA, which we call this 3-letter code. And when the polymerase comes here, it won't be able to go further because there is nothing to put, and then it falls off. And this creates also a condition where the reaction can now spontaneously dissolve and reform. So if you look at the animation, we could see that the primer will bind, the polymerase will actually synthesize it to a point, but it won't be able to go past the polymerase, stopper, and then it will result in a branch migration process where this strand will actually invade back and make it destabilize. So spontaneously, it will dissolve, it will rebind and debind. So this reaction actually happens isothermally, which means in one temperature and in a programmable manner. So we know the kinetics of this binding and extension, and we can design length of the sequences in this way. So we know that this strand here can at some point displace this elongating one, and then it can rebind and get extended again. So as part of this reaction, we can create long single-stranded DNA concatemers where the same unit sequence is repeated over and over. And this becomes essentially our amplifier concatemer for in situ signal amplification. This reaction is, highly controllable, so you can change the hairpin concentration, the polymerase or magnesium concentration to increase the length of these concatemers. Here we visualize it by putting a fluorescent molecule on the primer, so you can see how the length of the oligo changes as the reaction proceeds. And orthogonally, we have actually designed 50 amplifier molecules, so 50 different primer hairpin combinations, so we can use them for multiplexing. And so far, we have validated over 30 of them by in situ crosstalk assays, so we know that they can be used together and without crosstalk between each other. So this method we call signal amplification by exchange reaction, or in short, SABR. And if you want to apply it for immunostainings, we call it immunoSABR. In this case, we tag the antibody with DNA as before, and then actually generate this PR concatemer in vitro beforehand on test on a gel, then apply it for the in situ reaction, then apply the fluorescent imagers for detection, and then remove them. And we do this also in multiplex fashion, as I've explained before. So the single-stranded product and the linear product that we use is quite compact. It even allows us to do high-density labeling like these tubulin structures. And the sensitivity we get now with primary antibodies is, or the signal level, I would say, is close to or at the same level or higher than the secondary antibody fluorophore labeling. That's more typical. And the resolution is also comparable because these molecules seem to be quite compact. And the resolution we quantify by looking at what we know as full width half maximum of the signal. But in short, you can see that we are still in the diffraction limited regime when we do this. So in this case, we are using stable binding of fluorophores. So compared to the resolution approach with DNA paint I explained before, they are stably bound and imaged. We do not have this blinking behavior because not for every experiment you need this kind of high resolution. And it comes at a cost of course, doing much longer imaging times. So in this case, we didn't really need to do that. So, and to demonstrate the multiplexing, we have actually turned to mouse retina cryo sections. These are highly layered structures because there are different types of cell layers that are functionally also enriched in different cell types. So we can really clearly see the absence of crosstalk and the expected organization of the different layers. So we have looked at 10 different markers in this case with linear amplification, as I've explained before, and validated the approach to be feasible to be used in tissue samples. We're also talking about 40 micron thick tissue, so the penetration of the samples into this tissue was also quite satisfactory. We could also go one step further and actually use a branching strategy to increase or amplify the signal in an exponential manner. What I mean by this is that we can use, as before, a primary concatemer, but instead of putting the images directly on it, we can also use secondary concatenamers that bind a bit more stably than imagers here and create more binding sites. So this creates now the opportunity to even amplify the signal further in an almost exponential manner. So we call this approach branching, and you can even do multiple rounds of this to boost your signal. So if you look at how it works and compare the different levels of amplification, here we are looking at formalin-fixed paraffin-embedded human tonsil samples. This is zooming into one germinal center. And we are labeling a cell marker called proliferating cell marker called Ki67, which is expressed in the nucleus, actual nucleoli of the cells. In red, all the blue dots are DAPI. And in the unamplified case, we just left one imager binding site, or we did a linear amplification with one round, or branching, or even like iterative amplification with 3 rounds. And we can see how the signals, so here I'm showing them at 2 different contrast levels because it gets saturated at some point in one of them. So we can see how the signal now gets over amplified and amplified. This is in comparison to the more standard secondary antibody fluorophore imaging, which gave us about 8-fold, whereas with iterative amplification, we can go beyond 180-fold. We also compare this to a well-known amplification method called pyramidal amplification, or TSA. In this case, typically a primary antibody and a secondary antibody is used, and secondary antibody has an HRP, so horse peroxidase. The way this works is that the fluorophore is actually coupled to a molecule called tyramide. It's normally inactive, but in presence of HRP and hydrogen peroxide, it can be activated. And once it activates, it binds all the different tyrosine residues that's around in close vicinity of this complex. So it's a diffusive amplification strategy. You don't only label your target, but stuff around it. And one of the limitations of this is that the resolution is not so good afterwards because the signal dissipates. So we have tried a commercial kit to test this, and we can see that the amplification fold is really good, but the signal dissipates quite a lot. So the nuclear marker actually starts to go out of the nucleus. And in comparison, we could use iterative saber to go beyond this level of amplification, but still keep the signal in the expected compartments, in this case, nucleotide close. And if we actually apply it like we do on secondary antibodies with branching, we could get even more signal as well. So that made us very happy that this type of approach that we developed could be utilized also quite well compared to state-of-the-art amplification methods to deliver high signal with the potential of multiplexing and also better resolution and morphology. So this type of morphology, of course, preservation is important if you want to do automated image analysis later on, but also the high signal level really helps with throughput, because then you can image these whole sections of tissues in much shorter times. In this case, we have been combining linear amplification and branch amplification for different markers. So linear for more high expressed proteins like IgA, IgM, and branching for more lower expressed proteins like the immune cell markers CD8 and Ki67 again. So we can see pretty good resolution, but also we are able to get imaging times below 20 milliseconds. So these are exposure times for each of these markers, which makes the imaging of like really centimeter level tissue sections a lot more, let's say, feasible, and makes it the higher throughput. The same type of approach, I've shown it for protein, but this also can be applied for RNA or DNA detection. So in this case, we call it saberfish, and my colleagues Jocelyn Kishi, Brian Belliveau, Sylvain Lapin, and Emma West have been working on this front. And this is some of the applications from the retina, again, where 7 different markers were labeled. And you can see, again, the RNA distributions quite well. And this is another example of a 17-color walk over the human chromosome. So these are metaphase spreads, and 17 different orthogonal fish probe sets were utilized to walk across, sorry, the animation did not work there, to what you can see different colors to walk across the arm of the X chromosome here. So in this case, I'm missing an animation, but the way it works is that we have the barcoding sites on the fish probes directly, and we can actually extend the fish probes themselves into concatenamers and apply them with in the fish protocol, then followed by detection with fluorescent oligos. We have similarly also combined the SABRE approach with expansion microscopy to bring it to a sub-diffraction level for tissue samples. So expansion microscopy has been developed in Ed Boyden's group in Broad Institute a few years ago. And the way this works is that your tissue samples are embedded into a gel matrix, which many of the components of the tissue are digested, except the ones that you're really interested in. If it is RNA or protein, you actually fix them to a gel matrix. This is a hydrogel. So by putting more water in, you can actually expand this. It's like a baby diaper. And this allows to take the molecules apart from each other, so you can actually resolve them much better by just making the sample 4x bigger. So in this case, you can actually go below the diffraction limit by using a conventional microscope without using any other tricks. But of course, you also dilute your signal because the number of fluorophores that normally fall in 1 pixel are now also distributed. So we thought SABRE would be a good compensation for this, but also would make it a lot more feasible to do multiplexing. So in this case, we are staining the sample with DNA conjugated antibodies, and then we are amplifying with concatenamers, which are extended from acridite modified primers. And this is like the acridite modification allows us to cross-link the concatenamers to the gel. It's like a polyacrylamide gel when it's polymerized, and then it can link to the gel and expand with it. So this way we can actually resolve structures beyond the diffraction with the SABRE too. So this is a demonstration where we have these pre- and post-synaptic markers, Bassoon and Homer, which are normally overlapping because they would be in the synapse, which is like sub-diffraction size. But with expansion, we can actually start to resolve them. And this is also applicable in combination with tissues as well. And we can see at high zoom how now things could be a little bit more decluttered and separated better for the tissue samples as well. So overall, for this part, we have been developing a DNA nanotechnology toolset for multiplex and amplified imaging of proteins and nucleic acids. In this case, our workflow goes from designing in silico design of primers, barcodes, and probes, to then in vitro generation of things like the concatenamers and their quality controls, and then to in situ application for detection of proteins and RNAs or DNA molecules. And we aim to make it easy to multiplex and scale up. The amplification itself is programmable, but you do not use in situ enzymatic reactions, which could have other problems. And you can tune the amplification level for different proteins by using combinations of linear or a branch SABRE, and utilize standard equipment, even for high-resolution imaging with the expansion approach. So this toolset is being utilized by different groups now, or as well as us, in collaboration with different disease or medical groups. For example, for brain tissues, kidney tissues, we have recently actually just published a study in gastrointestinal stromal tumors, and we are trying to apply it in lung tissues for non-small cell lung carcinoma in collaboration with GSK as well. So, of course, now, what is next? So of this type of approaches, and there are other multiplexing approaches are great because we can look at many things at once, but they still don't have the depth of information that we could get from sequencing, especially next generation sequencing applications. So we were trying to next think of an approach where we can really interlink these together. So we can really utilize imaging for subcellular resolution and high sensitivity and get details of morphology, subcellular features, spatial organization. And on top of that, get to the level of sequencing depth where we would have a lot more high throughput and almost single cell level multiomics. So we can really study how subcellular spatial organization interfaces with functional cellular state by using newly developed computational methods. So for this, we have been working on a method recently, which actually just got published this week, which we call LightSeq for light-directed in situ barcoding of biomolecules in fixed cells and tissues for spatial index sequencing. So I want to talk a little bit about that in the remaining time. The approach we envisioned here actually goes from imaging, where we can use different markers for selecting regions of interest for in situ barcoding. And then we perform iterative barcoding rounds. So each of these areas have their own DNA barcodes. And then we can extract these barcodes and sequence with next generation sequencing methods from the pooled libraries, extract the region IDs and transcriptomic sequences, so we can do things like differential gene expression for each of these areas, and do this without destroying the sample, so we can even come back to the same sample and do follow-up assays like more immunofluorescence to validate our findings. So this was the vision that we had, and we had to come up with a lot of tricks to actually make it a reality. And so one of these is fast photo cross-linking for light-controlled in situ barcode incorporation. This allows us spatial selectivity. The second one was the one-step DNA stitching reaction that we had to engineer to convert spatial barcodes to sequenceable indices. And third one was to put all of these in a workflow for non-destructive extraction. So next, I want to go through each of these points and explain a little bit what I mean, so the method makes sense to all of you. But these are the three experimental elements that we had to come up to make this method go from images to transcriptome. So first, the fast photo cross-linking chemistry. In this case, we are using a nucleoside analog that is called 3-cyanovinyl carpazole, or in short CNVK. So this has a modification, but it could be synthetically incorporated into DNA oligos. And once it's there, when you have a DNA hybridization, it could actually create a cross-link. In this case, maybe it's to the -1 position across to a T or C residue. So it works much better with the T. So this is why we use it mainly for binding to Ts. And you need to do this with 365 or 375 nanometer illumination. It could actually work all the way to 405, but the efficiency is best around this UV illumination. In 1 to 10 second exposure, it actually, with very high efficiency, forms this cross-link, this photo-adduct to T bases. So this gives us a way to really fast photo cross-link DNA strands together in a light-controlled manner. And this is the trick we have been using for LightSeq. We have also utilized this approach before for actually doing super resolution level labeling in an approach we called Action Paint. So it's the same chemistry, but we were familiar with this approach, and we knew that it was very high efficiency. So we wanted to adopt it now for in situ labeling in a much broader fashion. So this is, we also call this DNA photolithography because it's really light-based incorporation of, let's say, DNA oligos in areas that we intend to actually label. You can imagine this if you were to do things in a tissue slide. Let's say we imagine that we coated this tissue slide with DNA oligos that have a particular sequence, for example, the sequence here. And then we hybridize our barcode sequences that are complementary to each of these, and they have this CNVK moiety in the binding site. So if we apply now like a patterned light illumination, we would only cross-link certain sites and not cross-link other sites that are not exposed to UV. And we can do a stringent wash to remove everything that is bound but not cross-linked, so that only the cross-linked strands will stay. So this gives us a way to really barcode spatially selected areas by exposing light to them. And this is the approach behind the spatially addressed labeling and the DNA photolithography that we utilized here. So, you can do this in vitro on surfaces, and we can do this in parallelized fashion by using a device called digital micromirror device, or in short, DMD. So you can actually, it's the same technology that is used in the projectors right now for watching TV or Netflix. So you can actually have many different small, tiny micron scale mirrors that can flip on and off so they could actually create custom patterns or photo masks on the fly. And this is much better than having a solid photo mask that you have to change every time. You can essentially custom generate photo masks. So you can apply this to any different photo structure. For example, this is a picture of some of the authors that are involved in this paper. And then we can convert this to a black and white mask and apply it onto the DMD and on a DNA surface to create micro-patterning with DNA. We could also do it for things like cat images in single color, or utilize this cyclic behavior to do multicolor. For example, this Penrose triangles model, we can actually encode it with 3 different docking sites, or 3 different barcodes in this case, to have different fluorophores incorporated for each of these, and then do a 3-color image of the DNA in the DNA space. So this is, of course, the strategy we are using it, but we want to apply it in situ on biological samples. And especially we wanted to be able to do this in fixed samples, because this is majority of the tissue samples we would have access to would be fixed rather than alive as well. So, you can create these type of photo masks now and apply it on cells. For example, this is like a checkerboard type of pattern where we can photo convert only certain sets of cells, or we can actually create micron scale dot arrays to label very small structures, custom select cells based on, for example, GFP expression, or custom select ROIs in tissues, regions of interest in the tissues to label. So this was more our test cases to see if we can use this approach for cells and tissues, and we validated that we could, and we could have pretty high selectivity spatially. So next we wanted to now see how we can use it for in situ barcoding so that we can actually use it for sequencing. So one of the problems is that when we do this type of reaction, we end up with a cross-link, right? So our barcode and the rest of the sequence we would have in the cell. And in this case, I will show you how it works, but we will have actually cDNAs that are in situ generated that has this barcode that we cannot really read out this by sequencing because there's a cross-link here. So we had to come up with a way we can synthesize a strand across this and create readout strands that would be in the right format for a sequencing library. So this is what we call cross-junction synthesis. So imagine we have a primer now to read this out. It will come here, but it will not be proceeding further. It's similar to the primer exchange reaction I explained before. We have changed the design such that there is internal complementarity here. So, the polymerase extends this to the cross site here, but it cannot go further. But we have designed this to be self-complementary such that at some point, this P strand here will actually dissociate and find another binding site here and could, dehybridize, hybridize, and then move over. So this is called a branch migration, and this is due to the competition between 2 identical domains that we engineered into the design of this barcode strands. So this allows actually, this strand to jump over, and then the polymerase to continue synthesizing all the way. And then we actually create the readout strand that has both the barcode region and then the transcriptomic readout sequence. So this is a bit complicated, but it works quite well with when you do a primer exchange type of reaction here, and we can recreate these single readout strands for sequencing. So the way now we put together all of the steps is you have your fixed and permeabilized sample where we have a fixed RNA molecule. We perform the reverse transcription reaction, which converts RNA to cDNA or complementary DNA in situ. So all of the RNAs we have are now converted to cDNAs. And for this reaction, we use a degenerate primer that's made up of like 5 Ns, so any nucleotide plus 3 Gs. And that has this barcode here, which we will use for bringing the CNVK barcodes. So we also apply something like a poly-A polymerase, so a non-templated polymerase that can add A residues here. So these cDNAs also have a handle on the 3' end, which we would use. Next, we perform these iterative rounds of light-directed barcoding with CNVK barcodes. as I've explained previously. And in between, we could also look at biomarkers, do protein stainings or other morphology stains to know which areas we want to barcode. So we can select these areas and apply different barcodes. After each round of barcoding, we wash the ones that are not cross-linked, so only the spatially selected areas are barcoded, and can add another orthogonal barcode. Here we can have different sequences to specify the spatial locations. After this, we perform the extraction procedure that is non-destructive by using an enzyme called RNase H. So this enzyme specifically cleaves RNA when there is a DNA-RNA hybrid. So like in here, we would actually cleave the RNA molecules, which will release these barcoded cDNAs, which we can collect from the sample. This will happen to all of the sample, of course. All of the RNAs will be released and degraded, and all cDNAs, barcoded and unbarcoded, will be released. But we can use these barcode sequence and the priming site that comes before that for specific enrichment of the barcoded cDNAs. But before that, we actually do the cross-junction synthesis using a primer that would bind to this part, and we'll go over, so the poly-A tail we generated on the cDNA that will jump over this cross-junction and put everything together into one readout strand. And then you can see these created here. And then in the next level, we do this PCR amplification to selectively amplify the barcoded cDNAs and perform the rest of the library prep and next generation sequencing. We have utilized it initially for visual pooling of co-cultured cells. For example, we mixed together GFP-expressing human cells with mouse T3 cells, and then we were able to select each of them separately by looking at microscopy images and introduce differential barcodes. So barcode 1 in magenta, barcode 2 in cyan here. And then by sequencing, we were able to resolve the mouse or human transcriptomes quite well. And we were able to do that just for 25 cells out of a whole well of these. In this case, these are well chambers, so slides with little chambers on them, which had 4,500 cells each. And we were able to assign the barcodes to human or mouse, or also eGFP, which were only present in the human cells as well, by using the mapped reads and resolving the different barcodes we put in by sequencing. We also have seen that because we use this degenerate priming or internal priming strategy, we are able to get many different types of RNAs, including, of course, mRNAs, but also link RNAs, mitochondrial RNAs, or non-coding RNAs as well. So this is also another advantage compared to some methods that utilize poly-A-based priming alone, which would only look at mostly mRNAs. Next, we wanted to see, okay, could we apply this in tissues for, let's say, regional transcriptomics? Again, we went back to retina samples, which we were most familiar with. And here we have these different layers again. So the ONL layer, which is really enriched in rods and cones cells, and BCL layer, so this is the bipolar cell layer, which is enriched in bipolar cells, as well as Müller glia and horizontal cells. And the third layer, the ganglion cell layer, which is really enriched in ganglion cells. We actually now created these types of masks manually, because you can see the layered organization of the retina even in a bright field image. And we wanted to see, could we really encode different cell layers with this spatial selection and get the sequencing information that confirms the different cell types that we see in these layers. You can see now this is the retina section after we introduced the barcodes. To visualize where we put the barcodes, for now we are also having fluorescent molecules that are different on each. You don't necessarily need to do that, but because these were the early stages of method validation, we thought that would be very helpful. And now we can visualize the specific incorporation into different layers as well. So afterwards, we applied the whole LightSeq protocol and then looked at our reads and classified them based on cell types. We could see that very nice enrichment of the barcode 1 in this ONL types of cells, then the barcode 2 in BCL and barcode 3 in GCL as expected. And we also were able to see all the different transcripts in the expected range, signifying the cell types. In total, we were able to actually capture 3,400 differentially enriched genes for the different layers. That showed that we are able to have a very good coverage of the different cell type markers that are expected differentially enriched based on single cell sequencing approaches. We also compared our approach with the existing drop-seq dataset. So this is an earlier method for single-cell RNA sequencing, which was applied to for the same layers in the retina. And in comparison, for looking at the BACL and ONL enriched transcripts, we were able to see that we correctly captured more than 98% of the genes that were previously covered by this drop-seq dataset as well. And we had good correlation for different replicates that also showed good reproducibility of the methods. So all the replicates were actually clustering together for different layers as expected. So this gave us a lot of hope that this method is applicable for spatial omics applications. And we also wanted to test, could we still come back to the same sample and do more studies? So here, this is the pre-sequencing barcoding, as I've shown before. And after this sequencing, we came back to the same sample, same exact section, and we performed either staining with, a lectin molecule, so it's called VTRM agglutinin, which labels the cell membranes, or two different immunofluorescent markers, PAC6 and VSX2, that we also saw in the transcriptome data to visualize, to be localizing into different layers. And we were able to immunostain them on the same sample and get good signal, which shows that in the protocol, our sample stains intact except for RNA and can be re-imaged as well. So this gives us a way to even come back to the same sample and perform post-sequencing staining. We haven't done too much multiplexing at this point, but it's completely feasible to do also utilize methods like immunosaber after sequencing as well on the same sample or before barcoding part to label the areas of interest. And finally, we wanted to now do a bit of a deeper study after performing these method validations. And we went for rare cell transcriptomics, where we really wanted to resolve a single cell subtype in the retina. In this case, we targeted dopaminergic amacrine cells, or DACs, which are quite challenging to also study with single sequencing methods, but you dissociate the cells because these cells are only about 0.01% of the retinal cells. So we were able to find 4 to 8 cells per whole mouse retina section. And they are quite hard to isolate. They are big and interspersed among many other diverse types of neurons, have these elongated morphologies. So they do not isolate well. But we know they are dopaminergic, so we know dopaminergic pathway markers should be there, and they can be labeled by one of the markers like tyrosine hydroxylase by immunofluorescence. So you can see that this is a DOPI-labeled retina cross-section, retina tissue, and we have 1, 2, 3, 4, 5, 6 of these cells with all of these processes that go across. So we thought, okay, can we actually get the transcriptome of these from these samples? And after performing the TH immunofluorescence, we then barcoded each of these cell bodies with one of our DNA barcodes, and we also barcoded and with orthogonally all the background cells, which are mixed populations of amacrine cells. So this is our TH label for the dopaminergic amacrine cell. Then we introduced the magenta, one of the barcodes, to all of the 6 here, and then the green barcode labels the mixed population that is forming our background. We then extracted these RNA sequences that we barcoded and performed the sequencing and differential gene expression analysis. So we were able to find actually 36 transcripts that were differentially enriched in the dopaminergic amacrine cells compared to the mixed groups. And then we went ahead on consecutive sections and validated these by performing fluorescent in situ hybridization using the saber-fish method I've explained before, so that we really can see that the ones we showed to be differentially enriched by sequencing are indeed found specifically in these cells. And we validated many transcripts that belong to the dopaminergic pathway, but on top, we found some interesting targets like CNC5, which is normally a voltage-gated sodium channel, very rarely expressed in renal populations, typically a cardiac transcript, as well as TRA6, a vitamin A transporter that has been implicated in retinal disease, but potential phenotypes that relate to dopaminergic amacrine cells was unknown. And these would be, of course, interesting points to follow going forward. We could also see, in more detail, these were the 36 transcripts we picked up that are expressed particularly in dopaminergic cells in enriched fashion compared to other amacrine cells. And this is another view of the fish labeling afterwards. We also have some non-differential controls that didn't come up, and that was like kind of everywhere, and we can see them also in these as negative controls here. And we were also able to come back and perform, as I've explained before, immunofluorescence stainings for some of them. So we revalidated that cells had a TH, and also looked at the CAR-TPT, another marker that we've identified for this population after sequencing in the same samples. For those of you who might be interested in how our results compare to other spatial omics methods, so we also quantified the number of unique molecular identifiers. So this corresponds to number of transcripts we detect. In this case, we were looking at 5 sections as technical replicates, and each of them had about 4 to 8 of these cells that we barcoded. So we went down to 4 cells per tissue, and we have quantified how many unique molecular identifiers or unique transcripts we identify. It was about 6 to 10,000 per cell. But these are large cells, so to also have a general measure, we quantified this for 10 micron by 10 micron unit area, which is again around the same range, so 6,000 to 10,000 transcripts that we were able to see. And this is in comparison to some of the other commercialized or available methods that utilized spatial barcoding, spatial decoding approaches, such as SLIDE-seq or DBit-seq, which were able to retrieve 1,000 or 5,000 unique molecular identifiers or unique transcripts per unit area of the same size. So although the results from different tissues could vary vastly, we think that we are on par or even better with comparison to these methods, and have a good coverage and depth for the transcriptome. So with this, the type of workflow we imagine for LIGHT-seq really goes from utilizing phenotypic characterization with subcellular multiplexed imaging. As I said, we can use DNA barcoded antibodies or morphology markers to do very high-level characterization. We can even use, if available, reference single cell data to select the markers. So we can focus on regions of interest that we want to sequence in the cell. And we can perform live image analysis to identify cells in these samples. So we can really define these ROIs, or regions of interest. Next, we can do the multiplex in situ barcoding for different regions with LIGHT-seq. And this would help us to identify cellular phenotype to gene expression links in a systemic fashion, and even go back to the same sample for further validation. We have been doing so far immunofluorescence, but we think some of the mass spectrometry assays could also be possible to do. So we are testing this out at the moment. So this type of approach has advantages that the sequencing reads are really concentrated only for cells or areas of interest. So you don't have to, if you don't need to, you don't have to sequence everything in your tissue to find a few cells of interest that you're particularly interested in. And that saves a lot on the cost side and sensitivity side as well. The approach is really compatible with different types of microscopy modalities. So you can use your favorite method for imaging and then find the ROIs and then do the barcoding. So you can, go to the subcellular level of detail as well, which is something we are very interested in. And the approach is quite accessible and affordable compared to some of the other very expensive spatial sequencing type of methods as well. So there are a bunch of potential applications. We have been now showing the capturing of rare or hard to isolate transcriptomes, but of course one could use LIGHT-seq to identify spatial heterogeneity and neighborhoods in cell subtypes, unmix the profiles from mixed samples, or even extend it to the level of doing perturbations in screens, capture interacting cells, including pathogens or microbial interactions. This is something we are quite interested in following up. And even do in situ microbiome type of studies, again, an area we are now focusing a little bit on. In addition to these applications, it could also be a great approach to create reference or validation maps for multimodal data from the same target cells without doing inference-based approaches, which is used to integrate many different multimodal data sets right now, because we can really go from same cells in the same tissue and do transcriptomics and imaging. So it will be one-to-one correlation. And the next level, we want to also push down the resolution. So from regional sequencing down to single cell sequencing. So in the current study, we were going down to let's say 4 or 5 cells per tissue, but we didn't encode single cells, like all the different single cells. So this is an area that can be explored further. But we are also interested in pushing it to subcellular level, because by using light, we can go down to the fraction limit. This is just to show the feasibility of labeling even subnuclear regions with our approach, so we could actually barcode this type of subcellular transcriptomes as well. And this is an area we are currently working on. Again, we could use protein markers on top by using DNA barcodes to add to the transcriptome information to do multi-omics level. This is something, again, worth exploring further. So with this type of approaches, we really want to bring together the imaging and sequencing methodologies and utilize the power of computational approaches to get down to the genotype to phenotype associations and patterns, and add all the richness of microscopy, from morphology to spatial organization to subcellular features, so we can understand cells and their interactions better. My current group in EMBL is following up some of these approaches. So we are developing new barcoding multiplexing tools and methods, and then we perform their applications for disease mechanisms and treatments, understanding resistance, or how we can actually find biomarkers. And we also try to integrate this with subcellular organization and figuring out the moonlighting activities of RNA and proteins, and see how they come together with disease mechanisms and potential treatment options. So with these type of approaches, we can really try to improve the depth of experiment, and this is one area we are pushing. We want to use them to understand how subcellular phenotypes and heterogeneity reflect the cellular state. How does subcellular organization contribute to the cell identity, functional state, and diseases? And if we can actually understand these better, we hope that we can reduce the cellular complexity to representative markers or parameters that would be predictive of or correlative to a cell's functional state, and use this information for biomedicine and pathology, from diagnosis to treatment, understanding response to developing new biomarkers. And so we can really understand better the color diversity in biology, especially the spatial complexity, and see what makes each cell unique and addressable as well. So with that, I want to finish thanking, of course, my group members. We are not too many yet, but has been growing in the past 2 years, as well as my collaborators, particularly the LightSeq contributors I've talked about, Jocelyn Kishi and Nining Liu, who were in Pangin's lab, and Constance Sepko's lab, especially Emma West, who helped us a lot with the retina work in general. And we are looking for potential new members, especially postdocs at the moment. So if you're interested, let me know too. And with that, I want to thank you for your attention, and happy to answer any questions. Okay, so I think I will just stop sharing so I can see you better.
Evet. Bu bilgi dolu sunum ve çok güzel anlatımınız için teşekkür ederiz hocam. Sizin de dediğiniz gibi o halde sorularımıza geçelim. İlk sorumuz, Unified Saber çerçevesinin protein ekspresyonunun fonksiyonel bilgileriyle single cell RNA sequencing analizini yapan araştırmacılar için faydalı olması ve aynı zamanda doku atlasları ve tümörler dahil geniş bir uygulama alanına hizmet edebilmesi mümkün müdür?
Evet, bahsettiğim gibi yani bazı tümör atlas ya da moleküler atlas tarzı programlarda yer alıyor. Saber mesela Human HubMap isimli bir program var, NIH'in organize ettiği. Oradaki tekniklerden bir tanesi olarak kullanılıyor. Şu anda biz amplification'a biraz yöneldiğimiz için özellikle transcription faktörleri gibi biraz daha zor olan signal amplification özellikle faydasının olduğu proteinler bizim için biraz daha ilginç. Böylece Saber'ı da diğer yöntemlerle beraber bir arada kullanıp daha da multiplexing artırabiliriz. Bu tarz uygulama alanlarımız var. Hem de patoloji için de güzel, özellikle uygun olduğunu düşünüyoruz.
Çok teşekkür ederiz hocam. Bir sonraki sorumuza devam ediyorum. Bölgelerini saplama potansiyelini arttırmak için hangi metotlar kullanılabilir?
Şu anda farklı eksplosyon. Evet. Eğer mesela LightSeq kullanmak istersek bir şekilde ilgilendiğimiz ya da özellikle analizini yapmak istediğimiz bölgeleri label'lamamız gerekiyor floresan moleküllerle. Bunun için normal işte morfoloji markerlarını kullanabiliriz ya da immunofluorescence yapabiliriz. Yani genelde bizim gördüğümüz en mantıklı yöntem öncelikle bir single cell sequencing datasından başlayıp hangi markerlar gerçekten önemli bu clusterları görebilmek için bunları kullanmak ve o yüzden de bazı mesela işte şeyde kullandığımız gibi tyrosine hydroxylase'i kullandığımız gibi retina çalışmasında bu tarz markerlar kullanılabilir. Ya da daha discovery odaklı gidilebilir. Belki genel markerlar kullanılıp machine learning ile bazı clusterlar seçilip bunlarda hangi transkriptomlar express olmuş diye bakılabilir. Hani daha böyle discovery tarzında kullanılabilir. Böyle çalışmalarımız da var. Aynı şekilde eğer in situ microbiome tarzı bir şeyle ilgileniyorsa bu sefer bakteriyel targetları label'lamak için markerlar kullanılabilir. Değişik opsiyonlarımız var. Şu anda hani en azından Light-Seq için metot biz immunofluorescence ve diğer türlü stainler mesela wheat germ agglutinin olabilir. Bizim kullandığımız ya da işte atıyorum MitoTracker vesaire değişik ya da ROS tarzı reaktif oksijen specieslerini görebildiğimiz markerlar olabilir. Bunlar kullanılabilir ya da morfolojik olarak işte şu alanla ilgileniyorum mesela tümörle immün sellerin interfeysiyle ilgileniyorum gibi morfolojik olarak distribution'lar ya da spatial organizasyon kullanılabilir.
Çok teşekkür ederiz hocam. Bir sonraki sorumuza devam ediyorum. İzole edilmesi zor hücre popülasyonlarının veya korunmuş dokulardaki nadir hücre tiplerinin görüntüleme bilgili, spatial olarak belirlenmiş, derin dizleme analizini tek bir cihazla gerçekleştirmenin Light-Seq ile artık mümkün olduğu bilinmekte. Peki bu tekniğin sınırlamaları nelerdir?
Çok güzel bir soru. Şu anda mesela üzerinde çalıştığımız bir şey sensitiviteyi arttırmak. Dediğim gibi biz aslında internal priming yapıyoruz. Bu demek oluyor ki degenerate bir primer kullanarak bir sürü değişik farklı RNA'yı alabiliriz. Ama aynı zamanda çok express olan mesela ribozomal RNA gibi RNA'leri de alıyoruz ve bunlar bir tür kontaminasyon olarak gözükebilir. Böyle durumlarda tabii sekanslama kütüphanemizin büyük bir kısmını bu tarz sekanslar alıyor. Bu da bizim sensitivity'mizi birazcık etkiliyor. Bu yüzden şimdi mesela bu kısmını düzeltmeye çalışıyoruz. Onlardan kurtularak çok daha yüksek bir sensitivity'ye ulaşmak istiyoruz. Aynı şekilde dediğim gibi single cell seviyesinde göstermedik. Subcellular seviyede de henüz göstermedik. Şu an bunları o seviyeye çekmeye çalışıyoruz. Yani resolution çözünürlüğü daha çok arttırmaya çalışıyoruz. Bunlar teoride mümkün ama pratikte biraz daha metodun yenilenmesini ya da işte küçük triklerin içine eklenmesini gerektiriyor. O yüzden bunlar şu anda üzerinde ulaştığımız alanlar. Mesela diğer metotlara göre belki coverage'ımız düşük olabilir, şu açıdan düşük olabilir. Eğer bir tuşudaki her şeyi birden map etmek istiyorsanız o zaman tabii bunu Light-Seq ile yapmak çok kolay değil. Çünkü çok fazla round yapmak gerekiyor. Onun yerine bütün doku örneğini disosiye edip toparlamak daha mantıklı olabilir. Ya da Visium diye mesela yeni bir yöntem var. Burada komple barkod edilmiş bir DNA ile barkod edilmiş bir slide'ın üzerine konuluyor doku örneği. Ondan sonra işte parçalanıyor ve RNA'lar o bölgelere tutunuyor. Böylece bütün dokunun bir genel organizasyonuyla ilgili bilgi sahibi oluyorsunuz. Eğer amaç buysa o zaman başka alternatif yöntemler tabii daha mantıklı olabilir. Ama biz biraz daha işte soruda da söylediği gibi daha rare sellere, az bulunan, ender bulunan hücrelere özellikle bu tarz sekanslama kütüphanelerinde kaybolan ya da çok net anlaşılamayan hücrelere yoğunlaşıyoruz. Bu da metodun ana güzelliklerinden bir tanesi.
Çok teşekkür ederiz hocam. Sıradaki sorumuz YouTube katılımcılarımızdan. Öncelikle teşekkür etmişler. DNA paint technique suffers from slow acquisitions due to low binding rate of the imagery standards, which methods can develop for this limitation?
Bu da güzel bir soru. DNA Paint'te açıkladığım gibi çok fazla aynı alanın görüntülemesini yapmak gerekiyor ki single molecule rekonstrüksiyonunu yapabilelim diye. Bunun bir de şeyleri çok uzun beklemek gerekiyor onların hepsinin bağlanmasını, geri bağlanmasını. Bir tane Fast Paint diye bir yöntem geliştirildi bir süre önce. Mesela bu olayı çok daha hızlandırıyor. Aslında burada yaptıkları bir nevi Saber tarzı bir yöntemle küçük konkatemerler kullanarak birkaç imagery bir arada koyup çok daha kısa sürelerde gerekli sinyali toplamak her molekül için, böylece çok daha hızlı olabiliyor. Yani bu fast paint mesela bu throughput'u arttırmak için kullanılan yöntemlerden bir tanesi. Alternatif single molecule detection yöntemleri de var. Storm Palm gibi. Bunların hepsinin tabii kendi limitation'ları ve avantajları var. DNA paint çok hızlı bir metot değil ama resolution olarak aslında 20 nanometrenin altına 5 nanometreye kadar inebilecek ender metotlardan bir tanesi ve yeni fast modaliteler var. Bu daha hızlı çalışan modalitelerle, özellikle bu küçük konkatemerlerin kullanıldığı, çok daha hızlı çalışabilir bir duruma geldi. Böylece daha pratik olarak da daha kullanışlı şu anda.
Çok teşekkür ederiz hocam. Bir sonraki sorumuza devam ediyorum. Konfigürasyonları ve girişimsel dış faktörleri düşündüğümüzde yerel yapı DNA'sının 3 boyutlu bir modelini nasıl yapabiliriz? Bu varsayılan model uzun vadede etkili olabilir mi? Yoksa her durum yedek bir modellemeye ihtiyaç duyar mı?
Burada soruyu bir daha görebilirsem, modelleme derken acaba hücredeki DNA'dan mı bahsediyor yoksa nanostructure'daki DNA'dan bahsediyor, çok emin değilim ama eğer DNA nanostructure'ları için güzel yöntemler var, computational metotlar var. Orada yani 2 boyutluyu 3 boyutluya taşımak tabii çok daha kolay, çünkü genel olarak 3 boyutun birbirine eşdeğer olduğunu düşünüyoruz. Eğer ki hücredeki DNA'dan bahsediyorsa bunun için çok daha farklı yöntemler var. Görüntüleme bazlı bazı yöntemler var ya da sekanslama bazı yöntemler var. Görüntüleme bazlı olanlarda genelde DNA tracing şeklinde yapılıyor. Bu gösterdiğim metaphase kromozomundaki bir örnek ama bunu daha high level'da yapmak lazım. Çünkü DNA structure'ları, oradaki 3 boyutlu yapılar çok küçük. Mesela TAD dediğimiz küçük loop'lardan vesaireden başlayıp çok daha büyük şeye gidebiliyorlar. Genelde bunu farklı domainlerde yapmak lazım ya da scale'lerde. Başka bir yöntemse onların yakın olduğu zamanlarda birbirine cross-link ederek bunu sekanslamaya çevirmek. Bu da high-C tarzı yöntemler var bunun için kullanılabilen. Hani hücre içindeki DNA organizasyonu için genelde bu tarz yöntemler şu anda kullanılıyor. Burada da öncü bayağı güzel gruplar var, çalışmalar yapan.
Çok teşekkür ederiz hocam. Özür dilerim. Chatten bir sorumuz gelmiş, onu okuyalım dilerseniz. Spatial çok hücreli model paternlerini ele alırsak, benzer hücre modellerini verimli, kısa sürede tespit etmek ve oluşturmak için bu paternleri hücre seviyesi özelliklerine nasıl indirgeyebiliriz?
Soruyu bir anlamaya çalışıyorum. Hücre seviyesi. Herhalde şeyden bahsediyor. Spatial çok hücre modelleri ele alırsak hücre seviyesindeki modellerle dokuyu modellemekten sanırım bahsediyor. Bu güzel bir soru. Bu bizim de çok aslında merak ettiğimiz, çünkü farklı seviyelerde yapılıyor araştırmalar. Gerçekten hasta ya da hayvan dokusu üzerinde yapılan araştırmalar var. Bir de bunları hücre modelleriyle modellemeye çalışıyoruz. Çünkü orada çok daha fazla perturb etme, olayı değiştirme ya da structural vesaire mekanizmalara girme şansımız var. O aradaki bağlantının ne kadar net olduğunu bilmiyoruz. Aslında biraz daha imaging based bazı molekül metotları kullanarak da bunu anlamaya da çalışıyoruz. Gördüğümüz feature'lar, sel modellerinde gördüğümüz feature'larla dokuda gördüğümüz feature'lar birbirlerini ne kadar represent ediyorlar. Bu ikisi arasında ne gibi farklar var? Hem bunları transkriptom seviyesinde hem de imaging depth, yani high resolution vesaire metotları kullanarak anlamaya çalışmak gerçekten önemli. Özellikle doku seviyesinde çok fazla high resolution çalışması yok. Yani subcellular resolution çalışması aslında çok fazla yapılmıyor. Çünkü scale çok büyük. Genelde tek hücreye ancak bakıyoruz. Yani tek hücre seviyesine ancak bakıyoruz. Ama biz işte biraz daha derine girerek bu aradaki boşluğu daha doldurmaya çalışıyoruz şu anda.
Çok teşekkür ederiz hocam. Bir sonraki sorumla devam ediyorum o zaman. Sabre Expansion Microscopy ile birleştirdiğimizde ulaşılabilir spatial çözünürlüğü sınırlayan düşük etiketleme yoğunluğu sorunu ile karşılaştınız mı? Çözüm için optime ettiğiniz koşullar var mıydı?
Evet, bu da önemli. Expansion mikroskopiyi değişik şekillerde yapmak mümkün. Bir tanesinde expansion'dan önce labeling yapılıyor. Diğer yöntemlerde ise önce expansion yapılıp sonrasında labeling yapabiliyor. Bu ikinci approach'un bazı avantajları var. Çünkü molekülleri birbirimizden ayırdığımız için çok daha fazla bağlanmaları için alan ayırıyoruz. Yani bu molecular crowding dediğimiz oradaki kalabalık ortam bizi daha az etkiliyor. Bu yöntemlerden bir tanesi olabilir ya da optimizasyondan bir tanesi olabilir. Tabii bunun da dezavantajları var. Çünkü artık label'larınızı küçük bir şey yerine 4 kat 5 kat büyüttüğünüz bir doku örneğine uygulamanız gerekiyor. O yüzden oradaki penetrasyon, difüzyon gibi şeyler, faktörleri de hesaba katmak lazım. Genelde çok daha uzun bir labeling protokolü uygulamak gerekiyor. Ama expansion'ın böyle ikinci bir artısı olabilir. Molekülleri birbirine uzaklaştırdığı için bu decrowding ya da kalabalıklığı azalttığı için şeyde çok daha labeling etkisi de çok daha iyi. Saber'ın da buna da fayda ettiğini düşünüyoruz. Çünkü bir molekül için çok daha fazla labellanabilecek site yarattığından dolayı o da daha düzenli labellama şansı tanıdığını düşünüyoruz genelde.
Çok teşekkür ederiz hocam. Sıradaki sorumuz YouTube katılımcılarımızdan. Mekansal analizde biyolojik bileşenleri kolayca ifade etmek ve aynı zamanda bu bileşenleri temsil eden veri setleri geliştirmek için çeşitli algoritmaların olması nasıl zorluklar açığa çıkarmaktadır?
Yani bu şu andaki daha bilgisayarlı yöntemlerde en çok karşılaşılan sorunlardan bir tanesi. Çok fazla değişik algoritma var ama o yani bu belki de asıl problem çok değişik algoritmanın olmasından ziyade datayı da kullanabileceğimiz çok fazla değişik yöntem var. Yani düzeltilmesi gereken çok fazla sorun var datalarla ilgili. Çünkü metotların çoğu çok yeni henüz kullanılıyor. Batch effect dediğimiz bir sample'dan diğer sample'a gördüğümüz çok fazla etki var ya da işte kullanıcıdan kullanıcıya gördüğümüz, metottan metoda gördüğümüz çok fazla farklılık var. Bunların üzerine tabii ki farklı algoritmaların olması da olayı biraz zorlaştırıyor ama bu tarz metotlar için daha çok yeni, yani alan çok yeni. O yüzden de bunlar normal. Yani 10 sene sonra genelde çok daha büyük bir standartizasyon göreceğimizi düşünüyoruz. Şu an gördüğümüz zorluklar mesela segmentasyon, hücreleri segment ederken değişik metotlar kullanılıyor. Bunlara göre tabii sonuçlar değişebiliyor. Farklı spatial metotlar için de farklı challenge'lar var mesela. In situ sequencing'deki decoding algoritmaları her zaman iyi çalışmayabiliyor. Hücrelerin ya da dokuların tipine göre bir çalışan şey başka bir doku için çok çalışamayabiliyor. Ya da yöntemlerin çok optimize edilmesi gerekiyor daha experimental'dan başlayarak. Tabii ki bu zorlukların hepsi farklı algoritmaların da kullanılmasını gerektiriyor bazen. Birazcık iki tarafında hem experimental hem de computation olarak standartizasyon zamanla olacaktır ama şu anda aslında bu varyasyon fena da değil. Çünkü experimental bazı problemleri çözmek için böyle bir varyasyona biraz ihtiyacımız var. Önümüzdeki dönemde daha framework structure'ları oluşacak yani ya da bununla ilgili çok fazla çalışmalar var. O yüzden bir standartizasyon, unification özellikle computational metotlarda bekliyoruz. Bu durumda da herhalde daha az algoritma, daha geniş uygulanabilir hale gelecek diye umuyorum.
Çok teşekkür ederiz hocam. Bu son sorumuzda aslında daha çok sorumuz var ama program akışından dolayı maalesef iletemiyoruz. Dileyen katılımcılar sorular için iletişim kurabilirler. Değerli vaktinizi ayırdınız, birikimlerinizi bizimle paylaştığınız bu bilim dolu bir saat için size çok teşekkür ediyorum. Katılımcılarımız ve kendim adına.
Ben çok teşekkür ederim. Benim için de güzel bir deneyim oldu. İlgilenenler tabii ki ulaşabilirler tekrar.
Çok teşekkürler. Sizi dinlemek için bizim için büyük bir zevkti. Görüşmek üzere.
İyi günler.